We all know how tired we get if we have a bad night of sleep. And if we have a couple of those in a row, it really gets exhausting. So can you imagine if you had to experience daytime sleepiness together with nighttime insomnia for months and years? That's what people with sleeping sickness experience. Sleeping sickness is actually an infectious disease caused by a parasite called Trypanosoma brucei. This parasite is transmitted by the bite of a fly called the tsetse fly that is endemic in 36 African countries. But this parasite is not picky. It not only is able to infect humans, as it's also able to infect wild and domestic animals. So it makes it not only a health problem in these countries, but because it affects their cattle, it also becomes an economic burden. But the World Health Organization has done big efforts towards the elimination of this infection, and the number of cases have been dramatically reduced. But there is still no vaccine against this parasitic infection. It's almost always lethal if left untreated. And in fact, the treatment itself is quite complicated. It requires intravenous treatment and is extremely toxic. It's believed that about 5% of the people that have this treatment die of complications. And this is all caused by a tiny parasite that is changing the sleep-wake cycles of its host. This parasite is not making these people sleep more, but instead is shifting the timing at when this sleep occurs. And the parasite does that by affecting the circadian clock of its host. But what do I mean by this circadian clock? Well, by living in our very rhythmic environment with day and night cycles due to the Earth rotation, organisms like us have evolved ways to anticipate these rhythmic changes. These are called our circadian rhythms. Our sleep-wake cycles are an example of those. We have hormones and some signals that make us feel sleepy during the nighttime, and the next morning, a different set of signals makes us feel active and hungry and ready to enjoy our day. These rhythms are repeated every day, and they last about 24 hours. That's exactly where the word comes from the Latin circadian, which literally means around the day. So these rhythms are also very interesting because they are endogenous. What this actually means is that they are not really driven by the presence of these light and dark cycles, but they are instead really coming from within. And this was shown by an experiment with plants. So plants have circadian rhythms on their leaf movement. The leaves go up during the daytime and they go down at night. And a French scientist thought that the plants were not sensing light and thus putting the leaves up, but instead that the plants were anticipating that the light was coming and placing their leaves. So he did a beautiful experiment, which consisted in putting one plant by the window that would be exposed to both day and night environmental changes. And a second plant was placed in a cabinet underneath it. So this plant was in complete darkness, isolated from any environmental cue. What he saw was that both plants, the one by the window and the one inside the cabinets, still maintained this leaf movement up and down. So this doesn't mean that light is not important. It's just it's not driving these rhythms, but it's very important to synchronize them. A simple way to imagine that is through jet lag. So I am originally from Portugal, but I live and work here in the US. But every time I travel to Europe to see my family, I have to travel about 6 hours difference in time zone. So my sleep-wake cycles take a couple of days to get synchronized to these new light and dark cycles. But light is important, and eventually I become synchronized with the environment in Portugal. So this means that we have circadian rhythms. Plants have circadian rhythms. But actually, circadian rhythms go all the way to the tree of life to the simpler organisms like bacteria. And since I'm so interested in parasitic and infectious disease, I wonder if this was just something that was common into organisms that are free-living and thus exposed to light and dark cycles. So my question is, do parasites also have circadian clocks? If organisms like parasites that are never really exposed to light and dark cycles, if they also have evolved this circadian rhythms mechanism. It turned out that the parasite I was studying, the sleeping sickness parasite, was a really good model to answer this question. And that's because this parasite is a single-cell parasite, but is also extracellular. So as you can see, we can culture it in the lab, and it's by itself, so no host cells involved. So whatever rhythms I can identify, they're really coming from the parasite and not from the host. But where to start? Well, I cannot just go and look at the leaf movement in this parasite, right? So instead, I had to take an unbiased approach and look at gene expression in these parasites, this gene expression that can potentially regulate all the downstream rhythmic behaviors. So in order to do that, I would have to first synchronize the parasite population to day and night cycles, just like the light and day for us. But for parasites, I thought that they might not be really responsive to light. They are actually always inside our body, so they are not really exposed to it. But instead, they may be exposed to temperature fluctuations. Our temperature drops at night when we are sleeping, and it increases during the day when we are awake and active. So parasites that are swimming in our blood could potentially sense these temperature cycles. So to mimic those, I actually cultured parasites and shifted every 12 hours in between incubators, so they would be exposed to day and night temperature rhythms. Afterwards, I either kept them in these alternating conditions or placed them always at 37. So in the end, I was trying to replicate the experiment that the French scientists did with the plants either by the window, exposed to day and night cycles, or at always 37, which will be the plant inside the cabinet. Okay, I tried to synchronize my parasite population, so now I'm going to go and check what are their gene expression. But I don't know what genes to look for. So instead, taking this advantage of a technique used in biology called RNA sequencing, I'll be able to not only count the number of molecules of RNA being expressed through time, but also to which proteins they can potentially encode. So this can be simply explained here by this diagram. As you can see, at 0 hours, this plant has only 2 molecules of RNA, one that is red, one that is blue. But at 12 hours, the blue molecules are now 4. So if I plot this data, I can actually identify that the red RNA encodes for a protein called protein A, and thus the RNA is called RNA A, and it's constant through time. Whereas the RNA blue, it actually has a higher expression at 12 hours, and then it drops again. So this actually has these rhythms in gene expression. So when I take a look at what are the gene expression in my parasites, I'll be specifically searching for genes that have the gene expression pattern like this RNA blue. So when I first look at what happened in the parasite population that was alternated between these incubators, I was very happy to see that we were able to synchronize the parasite population. And that's because we indeed found genes that were cycling, like here, this gene A. So you can see that the gene was highly expressed first, and then it got dropped, and then it went up again. And this was repeated on these 2 days that I measured. And this was not the case of only this gene A, but also for these 1,500 genes. And actually here in this plot, the genes are representing 1 row for each. And the color code represents their gene expression level. So you can see that in the beginning, for example, gene A is marked as red, and then it becomes blue, and then it gets red again, and blue, and so on, which will be an example of here, one of the few first rows in this heat map plot. So this was what happened when the temperature cycles were present. So I cannot really be sure if these are circadian rhythms of the parasite or just genes that are cycling because they are responding to the temperature fluctuations. So instead, to really be sure that parasites have circadian rhythms, we have to look at what the gene expression of the parasites inside our 37 incubator look like. So, exciting enough, parasites had gene expression cycling even in the constant conditions, even in the absence of any environmental signals. And this was not only for gene X, but actually for 1,000 genes that were cycling in these constant conditions. So these are indeed circadian rhythms, the really endogenous rhythms coming from this Trypanosoma brucei parasite. So this was very exciting. This really proved that parasites can indeed have circadian clocks. But the question now is, what are these clocks for? Why are the parasites putting so much effort in making 1,000 genes cycling throughout the day? What are these rhythms actually encoding for? So to answer this question, we went and specifically looked at what are the genes that are cycling throughout the day. And what we found was that most of these genes are encoding for metabolic pathways. They are either involved in the metabolism of sugars or carbohydrates. They're involved in the metabolism of lipids or fats or in the amino acids of proteins. But this was all at the gene expression level. So I needed to validate if this was really true for the parasite population, if the parasites were somehow in a different metabolic status throughout the day. So I could potentially go and just look for the expression of these proteins, but instead I looked at ATP levels. So ATP can be seen as the ultimate energy molecule that will be results of all these metabolisms. So when I measured the ATPs in these parasites, I found that the ATP levels are going up and down throughout the day. And in fact, this happened in the conditions that were in alternating incubators, but also in the condition that's always at 37. So you can also see that the timing of these higher ATP concentration also matches the higher that you have more genes being expressed of these metabolic pathways. So there seems to be a big importance of circadian rhythms into regulating the metabolism of these parasites. This was very interesting because, of course, I'm very passionate in understanding how the biology of these parasites work. But also, in the end, I'm interested in trying to find ways to fight them. And because we know that most of the anti-parasite drugs, they actually act on these metabolic pathways. It could be that just by simply administering the drug at specific times of the day, we might find the parasites more susceptible to this treatment. So I set out exactly to test this question, to try to answer it. And I first started by synchronizing this parasite population again with our temperature cycles. Then, to each of these cultures, I gave an increasingly higher drug concentration and hoped for the increasing higher drug to kill more parasites. And that's exactly what I saw. So if I give very little or no drug, 100% of the parasites remain alive. But as I increase the drug concentration, these parasites start dying. And this was a drug called suramine, which in fact is known to act upon the glycolytic pathway. And it's used to treat sleeping sickness parasites in the field. So this was very exciting. The assay was working. So I could then ask whether this exact death of parasites through this suramine drug was similar throughout the day. So when I looked at what happened at 4 hours, I was very disappointed because it was exactly the same. You can see that these curves look the same whether I treat parasites at 0 hours or whether I treat them at 4 hours. So this could have just meant that my hypothesis was wrong altogether, that there are no benefits in treating parasites at different times of the day and that their metabolic pathways changing is not really correlated with the treatment. But instead, I didn't take that as a no because these are just 2 time points in the 24-hour day. So instead, I went and looked at what happened at 8 hours. And as you can see, I was extremely excited because there is now a shift. Now parasites are requiring higher drug concentration in order to be killed. So for killing the same amount of parasites, now the drug needs to be at higher dose. This was very exciting, and so when I finished for all the time points throughout the day, I can then transform this plot into the dose that I require to treat half of the parasite population. So you can see here that if I treat at 0 or 4 hours, I need half of the amount of drug to kill the same number of parasites. So if I treat them at 8 hours instead, I would need 2.5 times more drug. And this really means that there is a potential for improving the treatment by simply administering at different times of the day. So altogether, I was able to show that Trypanosoma brucei parasites have circadian rhythms, and that these rhythms regulate the parasite metabolism, making it more susceptible to drug treatments at different times of the day. But as you can imagine, parasites probably didn't evolve these circadian rhythms in order for us to use it against them. I can think that parasites have evolved these rhythms to anticipate when the vector is coming to bite at the end of the day, to be prepared and thus enhance transmission. But I can also think that maybe parasites have evolved circadian rhythms in order to predict when the strong immune response is going to happen, because our immune system is also cycling throughout the day. I can think that parasites may have evolved circadian rhythms in order to anticipate when the nutrient influx is going to happen. We only eat during the day, right? So there are many different reasons for why circadian rhythms may be so important for parasites. But what we know now is that they have them. Trypanosoma brucei has circadian rhythms, and there is a potential to use the metabolic rhythms that it has to treat them at specific times of the day. So I think it's worth to confirm that our results from the cultures really translate into differences in mouse models. And what we found for suramine may be also true for other different drugs available to treat this infection. Also, it's worth for us to go and look at what are the genes that actually regulate these circadian rhythms. What are the few genes that regulate the expression of these cycling 1000 genes? And if we identify them, can we actually use these genes as potential drug targets to disrupt the circadian rhythms of these parasites? Finally, what about other parasites? So do parasites have clock? It's a very general question. We should look at what happens in circadian rhythms in malaria parasites, in leishmania, in toxoplasma, all these other infections that are very bad for both humans and animals. Do these parasites also have circadian clocks, and can we use it against them?